Hi, everyone. My name is Quincy. I'm a second-year PhD student in Bioinformatics and Computational Biology program, trained jointly with the University of Minnesota Twin Cities and the Mayo Clinic College of Medicine and Sciences. Today, I'm happy to present a poster that talks about using deep learning to automate breast cancer diagnosis from hematoxylin and urisin, which is H&E stained microscopy images. Breast cancer is the most common cancer in women. Non-metastatic breast cancer with early diagnosis is highly curable. H&E stained breast biopsy slides are the gold standard for pathologists to diagnose breast cancer. However, the concordance rate in interpreting breast biopsies is unacceptably low. Since deep learning has successfully applied in medical image processing field, we evaluate supervised deep learning-based image classification models for differentiating malignant from normal breast tissues. As choice of hyperparameters is well known to affect deep learning models' performance, we also compared several different hyperparameter configurations. The data we use for the study are H&E stained breast histology microscopy images from the Grand Challenge on Breast Cancer Histology Images dataset, which I'll be referring to as the BARC dataset. Since the objective of the study is a binary classification problem, we label the images by assigning images from the normal and benign classes as a class 0, and assigning images from the in situ and invasive carcinoma classes as a class 1. Then we split images into training, validation, and testing sets for the model training and evaluation purposes. We implemented our models in TensorFlow. We tested the image patches on two groups of deep learning-based image classifiers. The first are general image classifiers, which includes one-shot learning and transfer learning with Inception v3, density 201, and ResNet 152 architectures. The second is digital pathology specific group, which only includes the clustering-constrained attention multiple instance learning, also known as the CLAM model. CLAM required an additional feature extraction step using a pre-trained ResNet 50 model on the ImageNet dataset for lower dimensional representation of the images. However, two additional problems remained. The original implementation of CLAM was in PyTorch. However, the general models were TensorFlow-based and did not accept TIFF images as inputs. So we implemented CLAM model in TensorFlow with support for TIFF images. To ensure our implementation performs similar to the original CLAM model, we compared the performance of the two CLAM models on 40 TCGA breast H&E slides. We found our implementation performed similar to the original CLAM using a student's t-test, where the p-value was 0.67, confirming that the results are concordant. We then tested all 5 models, showing table 1 of this poster, with multiple combinations of different hyperparameter configuration, showing table 2. We found that one-shot learning and density 201-based transfer learning with highly tuned hyperparameters are the best performing models with 94% classification accuracy on the validation dataset. Based on the results of the study, we found that combination of hyperparameter configurations have a direct impact on the performance of deep neural networks for image classification. Therefore, to identify well-performing deep neural networks to classify tumor versus normal breast histology, we highly recommended researchers to not only focus on devising new models specifically for digital pathology, but to do extensive hyperparameter tuning for existing deep neural networks first, as this also achieve a high prediction accuracy. Thanks for listening.